Hello, I'm Carolina Dace Cruz, a senior biology major at the University of Puerto Rico in Rio Piedras. For the past year and a half, I have worked in Dr. Alfredo Gessi's lab as an undergraduate researcher. Dr. Gessi's lab studies the molecular basis of neural adaptation. My project investigates the epigenetic mechanisms behind alcohol tolerance, specifically the mechanisms that alter gene expression and chromatin structure. Researchers know that alcohol use disorder causes long-lasting neuroadaptive changes in the brain that are expressed as tolerance and physiological dependence, both of which promote the addictive behaviors. Previous studies have shown that histone modifications may be involved in mediating these changes that occur during ethanol exposure. Acetylation and deacetylation of chromatin regions have been shown to control several aspects of the alcohol response, including tolerance. However, these mechanisms are still poorly understood. This project aims to understand the mechanisms underlying the changes in the brain that lead to compulsive and escalated alcohol consumption. We specifically investigate if the histone acetyltransferase Tip60 plays a role in the development of alcohol tolerance. To test this, we divided the flies into the control group and the experimental group. The experimental group consisted of Tip60 knockdown flies. During the first exposure, the experimental group was sedated with ethanol using a vapor chamber, while the control group was left untreated. For the second exposure, flies from both groups were sedated with ethanol. The locomotor activity for both groups was measured using a Drosophila activity monitor. The time of sedation was compared between these groups to determine if tolerance was acquired. We observed that Tip60 knockdown flies had a reduced capacity for alcohol tolerance. They did not demonstrate a significant increase in sedation time between exposures. The control group, in contrast, showed a strong tolerance. This suggests that Tip60 is necessary to acquire alcohol tolerance. This finding helps us better understand how the brain adapts at the molecular level to alcohol and the mechanisms that contribute to the maintenance of addictive processes.